So we've loaded our protein separation gel. They look really good. Remember, we're using lanes 3 through 9. We're using lanes 3 through 9, and the gel that we are running these in is made out of a substance called polyacrylamide. And it's later, when we take these apart to do the blotting, you'll get to kind of feel it, and it's more rubbery than a DNA gel, much more solid, and extremely thin. So right now, these proteins are separating through the gel. They're moving toward the positive electrode down here because they have a negative charge. And they're moving right now through this stacking gel, which is low percentage polyacrylamide. So they're going to go pretty fast. And then when they hit this boundary and go into the high percent polyacrylamide, then they really start to separate from each other. Which proteins are going to go the furthest in here? The smaller they are. Yeah, the smaller they are, the further they will go, and the faster they will travel. So we're going to run these until we see the dye front, which is that blue dye that is in the samples. The dye molecules are smaller than any of the proteins, and they're what goes the fastest. So you can use this dye front and watch it move through the gel, and when it gets near the bottom, then you know it's done. We're actually using 30 minutes, regardless of where the dye front is at that time. So once we have separated the proteins, we will have mostly invisible proteins separated in this gel. We're not going to be able to see any of them except for the ladder protein. Except for the ladder protein. And maybe the actin-myosin standard. So the ladder that we loaded in lane 3, it's got fake colors put onto the protein band so that you can see where they are. And we'll be able to see those in the gel. But everything that came out of our seafood samples, there's going to be proteins there, but they're going to be invisible. You have to trust that they're there. Here's something that's going to be, I didn't know until this week. So we've got 4 gels right now. One of them is mine, and then your 3 groups. We actually can only blot 2 of them because we have the equipment to blot 2 of them. So what we're going to do is we're going to do Western blots on 2 of them. The other 2, we're going to do a direct stain on those. The stain is just a dye that we put on the gel itself to make the invisible proteins visible. So 2 gels will get directly dyed by putting stain onto the gels. The other 2 gels will get blotted. And probably the way I'm going to do this is whichever ones look the best, we'll take those and blot them, and then the other 2 will stain them. And you'll end up having a picture of a stain gel, and you'll put that in your notebook. Even if that wasn't your gel, it's a good example. And at the end of blotting and detection, we'll have a picture of the actual blot where we detect muscle proteins. So we'll see how those look to make that decision. These, the ones that we're going to blot, let me start with these. The ones that we're going to stain, all you really do for that is you soak in buffer, and you soak in something called Kumasi stain. And the Kumasi stain kind of has a brownish bluish color to it. It stains the proteins blue. It kind of acts like Bradford dye a few weeks ago when we did protein assay. So this Kumasi stain is going to, in the gels that we use it on, it will color every protein that's present in the gel. It will be blue. It will be blue bands. So the whole gel will look like a bunch of blue bands with this kaleidoscope ladder having its individual colors. And this will show you, this shows you all proteins in the gel, no matter what their identity is. If it's a protein and it's present in a reasonable amount, you'll see it in a stained gel. Okay, so now I'm going to go and talk about the blotting procedure. And the blotting procedure, you actually have it in your packet. You can follow along kind of there. So when we do the blotting, what we're trying to do, the purpose of this, the purpose is to move proteins out of the gel and onto a membrane. And if we have the proteins on the membrane, we'll have easier detection in step 4. So we're going from proteins in the gel, get them onto a membrane. This particular membrane that we use is made out of a chemical called nitrocellulose. So it's kind of like paper. That's the cellulose part. It's kind of like paper. It's got chemical modifications to it to make it charged, in essence. I think it has a positive charge, so you pick up negative proteins onto this membrane and they stay stuck. So if this is our membrane, our nitrocellulose, we're going to have our gel laying on top of it, which is going to have bands, which are invisible, but we trust that they're there. And we're going to use electric current, again moving from negative to positive, to move those bands, those proteins out of the gel and onto the membrane. And then you can kind of imagine them like they stay on the surface of the membrane. Like if you squashed bugs onto the floor. They don't sink down into the floor, they stay on the surface. And proteins on the membrane can be detected by antibodies. We can take this membrane and float antibodies on its surface, and specific antibodies will bind to specific proteins. But this process, getting proteins out of the gel and onto the membrane, this is not specific. So we'll get ladder proteins onto the membrane, if this works well, plus all the invisible ones in our fish, seafood. It's a bulk transfer out of gel onto membrane. And the membrane is nitrocellulose. I want to pass out now we're going to do 2 blots, so I'm going to pass around a couple of sets of membranes. And when you order these, they come in packaging that sandwiches the membrane between pieces of paper. In this particular company that we get ours from, the paper surrounding the membranes is yellow. I better not get chalk on it. I'll just pass around one of these. So if you're in a group that does blotting, you've got yellow paper. That's not the membrane. That's just to keep them separate from each other. And then between there's a shiny, looks like shiny paper. So when you handle it, you're not supposed to touch it with your hands. It's coming between yellow cushioning papers, I guess, I would say. So just kind of see how that's oriented. And then the other thing, if you're in a blotting group, that you will get is filter paper. And this is just more like thick, cushy expensive paper. It's not particularly special. You can touch it with your hands, and it just has like a lot of thickness to it, and it soaks up liquid. So if you're in a blotting group, you'll get, I'm not passing these around because they're not that amazing. It's just thick paper. Did you touch it? Don't touch it with your finger. Because guess what will happen? Proteins from your finger go onto it. Yeah, you should actually, when you do this next work, you should wear gloves, yes. Turn to page 53 in your packet.
Dr. Ron, where do you want the nitrocellulose?
Back in my hand.
Whatever.
Yeah, nitrocellulose. I didn't, Becca, who is our lab coordinator, she's like very responsible about putting things where they go. These are technically flammable. So she made a note, in the flammable cabinet. Like, really? Don't light them on fire. They're flammable from the nitro, I suppose.
I like how you said they're technically flammable.
Well, like, it's not like they're going to combust spontaneously. Flammable. Okay, so when we're going to get the proteins out of the gel onto the membrane, this is an electrically driven process. And this picture in your packet is showing what is the order of this stack that you do to allow this process. So this blotting process requires liquid. You have to have liquid to help carry the proteins toward the positive anode. So when you're assembling this stuff, you have to assemble it in liquid. So you're going to be making this stack of layers, and you have to keep all the layers wet and put them in the exact correct order. So if you're in a blotting group, we're going to use these stain trays. These are a good size to do the assembly. Every layer that you put in there needs to be soaked in the buffer. So we'll be doing the assembly in the trays. The other part of this, so when you look at anode, cathode, those are part of the machine. We actually use the same gel box to do the electrical transfer. We just put a different holder in there. So that's part of the gel machine. The next things where it says gel holder, those are a specific apparatus, a specific equipment to hold this stack together tight. So these are quite cool. They come apart like this, and I think you start with black down. Yeah. If you're in the blot group, you're going to start this process with black face of this down in the bottom. And then that helps you get oriented in the electrical field later. So you have, this is the gel holder, black side down. Then you're going to have paper, the paper that's only moderately special, the filter paper. Then you're going to have the gel laid on top of that. You have to roll the bubbles out. We're going to have test tubes to roll the bubbles out so that there's no air gaps between any layers. Then you're going to put the membrane. It works best if you lay the membrane down like this, like let it kind of roll down, and then you'll roll the bubbles out. I said gel, right?
Yes.
Did I say gel? Okay. Gel, then membrane, then another paper, and then on the top something called a fiber pad. So I actually skipped over the fiber pad on the bottom. So this would be like a super Oreo, actually. So you've got fiber pads on the outside. These are just liquid holders, basically. They're these. So fiber pads, paper next to those, and then in the very middle, membrane on top of the gel. And all of this has to be black side of the holder down. So when you're assembling, if you're in a block group, make sure that you are literally getting these layers right. It's really important. So when you do those layers, you're going to close up. You close up this thing so it's got a clip that goes over and slides, and that will mush everything together. And then those go into this holder. So the reason we can only blot 2 gels is we've got one holder, we've got one gel box. It holds 2. If this goes well, next year we'll get another one. Let's test this out. These go in like this, black to black, with the correct electrical orientation. And then we run current through this, and that will pull, the current will pull proteins out of the gel onto the membrane. Here's another important aspect of this. When you lay the gel, let me go back to this part. When you're getting ready to put the gel into this stack, you have to cut off the stacking gel. And it says to do this with a plastic ruler. There's some of those in these drawers that you can use. So you need to cut that off. And then you need to cut off the die front down at the bottom also. And then when you do this, it's important to know what's the orientation of my gel. We're going to have a pre-stained ladder to tell us that. Like the ladder is going to say, oh, this was this side of the gel over here, and the ladder ought to transfer onto the membrane. But just to make sure, I also would like for you to cut that corner of the gel as well. Just for extra, like knowing which side is which, and what's left and what's up and all of that. So make 3 cuts. 1, 2, cut the corner up there. Because when you lay the membrane on here, the membrane is face down. And if this blots correctly, now I gotta get these in order. Your ladder is going to come off on the other side because the membrane was laying face down. See how that's going to be?
What's the bottom part?
The bottom part?
Yeah, where you drew that line, what did you call that?
The die front.
That.
Yeah, so you want to cut the die front off before you put this gel into the membrane stack. So you shouldn't have blue dye come over onto your membrane because that should have been cut off already. And then the order of your lanes, so this was lane 3, like it was lane 3 over here, and then it will be 4, 5, and so on, going the opposite direction. So don't be surprised when that happens. That's supposed to happen. I think that used to bother me so much that I loaded my gels backward so that my picture would come out left to right like we read. Oh well. That's fine. Okay, turn over to your protocol now, which starts on page 54. If you end up in a staining group, I will go through with your group what you need to do for the staining. Assuming that you're going to be in a blotting group, actually both groups are going to do the chopping, the cutting off of the gel. So no matter what group you're in, you need to cut off the stacking gel, cut off the die front, make the notch, no matter which group you're in. And then if you're in the blotting group, you're going to get, actually everybody's going to have these small trays. And there where it says on page 55, equilibrate the gel, you're going to fill up a little bit of blotting buffer in this little tray. And the blotting buffer is over here. You just come and get some. It's labeled blotting buffer. Pour some in the tray and then put your gel into it. And the gel needs to soak for 15 minutes. If we do not make it with all of this by the time class is over, I will do this stuff, but we might make it. So you'll kind of, anytime you've got to soak the gel or soak the membrane or let it equilibrate or wash, we're going to use these little clear trays for that. They're a good size for that. While that's happening, you get your silver tray and soak your fiber pads in it, just to make sure they're totally soaked with blotting buffer. It says that you need to put your initials on the nitrocellulose with a pencil. So put something so you know which group is which on your membrane. Then you're going to assemble the sandwich in this thing with the holder black side down. You got to get the air bubbles out with a test tube, and then you seal it up. It will go into this thing, and then we will run it for 2.5 hours at 20 volts. 20 volts. Does that seem okay?
The gel, like, it's on that tray right there, like the clear one. You like transfer it once you're ready to like flip it
Yeah, once you want to cut off the pieces before you put it in here.
on. So, but once you're, after 15 minutes, once you're ready to like set this all up.
Oh yeah, what you will do then is you're going to pour off. You'll pour off the buffer. Try not to let the gel fall out. So I just be careful. We pour off the buffer, and then you pour in the next thing. I think on this step you don't have to pour off the buffer actually. You just soak it in buffer, then it goes into the blot. It's the next round of things where we keep changing the liquids that it's soaking in. Something else. Something else that we have to do that is a little bit difficult, that's not listed on here. It might be back in your previous packet about running the gels. Yeah, page 49 in your first packet. We have to get the gels out from between the plates, and this requires you have to put some tweezers in between those plastic plates and break them apart and then get the gel off. So everybody is going to get some metal forceps or tweezers to help you get in between the plates, break the seal, open it up, and then you can chop the gel with your plastic ruler or, yeah. We should find plastic rulers now and get ready. Like here's one. This is, one group can use this. They're like this. So now we've got 2, we've got 3. Good. Okay.